Phylogeny. So back in 1837, in Charles Darwin's notebook, he drew this image here, which kind of looks like a tree with different branches on it. He wrote, "I think," beside it. And this essentially was the first phylogenetic tree. So we're going to look at phylogeny today and how phylogenetic trees are built and how they show the evolutionary relationship between species. Now you can imagine if you built more and more and more branches to the tree, the space of the branches are going to take up more and more room. And this here is a modern phylogenetic tree, sort of the next step. This shows 3,000 different species relationship, going back to something that they were related to, and then going out and branching as it goes along. We're going to look at how that works. So this example is taken from the Tree of Life, so you can check it out online. But essentially it's a way of trying to take our concept of time from a relatively short time frame into a much longer time frame that is necessary to fully understand how these phylogenetic trees work. So we know the genetic relationships between parents and their offspring, so that's easy to imagine, sort of family tree type thing. But as we go further and further back in time, these distant genetic relationships become harder to imagine. So we want to get to the point where we can see the relationship between things like daisies and elephants as long as we go far enough back. So this particular example uses a beetle, which is named Mimi, and Mimi species reproduces sexually, so it has a mother and a father parent that gets its copies of DNA from. Let's look at how this would look. So here's Mimi, and she has a parent and two parents. So let's say one mother and one father, and they both pass on genetic information. That is what Mimi uses in her genetic code. And there's some other beetles that are Mimi's siblings that are also descendants from these two parents. So sort of a family tree type of thing with two parents and three siblings in the next generation. So one generation, not too hard to imagine. If you go to two generations, and there's some more complex relationships here, but you can imagine here's Mimi right here. Let me get a different color so we can see this better. So here's Mimi right here, and there's her sibling and the sibling. So that means this is her parent and this is her parent. But her mother also had some other children with another father, and they are Mimi's half siblings. And then if we go back, so there's her mother's, one of her mother's parents, and there's one of her other mother's parents, and there's one of her father's parents, and there's one of her other father's parents. So these four here are her grandparents. And so this tracks the relationship through 3 generations of beetles. Not too hard to imagine still. And what you can see is that within generations you start having more of these connections between the different generations. Alright, here is a much more zoomed out view. So here we have all of these generations. I don't know how many there are, but a lot more than 3. And over time, so as you go back in time, we're getting more and more and more generations. And so the dots represent the individual beetles. There's Mimi. And the green lines represent the potential relationships or interconnectedness between those generations. So we can start to see that, okay, well, this is going to have a lot of different relationships going through the different generations. But you would be able to track Mimi's lineage, but again, as it went back, you'd have more and more and more connections because she's going to have more ancestors the further back you go. So that is going to be several generations there. Now, if we zoom out again, we can imagine that whole box that we were looking at before is just this one little part here. So if we go back further and further and further, so not just like a few years, but many, 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 many years, this lineage of beetles is going to get relatively long. The longer we go, the longer it goes. But we can actually go back to the point where down here was a group of beetles in which some of them diverged this way and became Mimi's type of beetle, and some of them diverged this way and became this other beetle. And as long as we don't have the two populations interacting, that divergence can take place. And over a long enough period of time, because those two populations are separated, these two beetles end up with slight differences. And if they end up different enough that you can't breed them and produce fertile offspring, then they're defined as a new species. Now, if we keep going back, so that's one divergent, but if we keep going back further and further and further, so here's those two beetles. So if we keep going back further, that's what we saw on the last slide, and if we keep going back further and further, that could have happened again. And so we would have had this lineage that led to that type of beetle, and this lineage that led to Mimi's type and the other type, two different species, but they're closely related because they didn't diverge till, I don't know, 1.7 million years ago. But when you go back to these guys, their most recent commonality that they had was about 3 million years ago. And you can go back even further. So Mimi is related to these beetles over here, but it was so long ago that that split happened. So we'd have to say, okay, well, that could have happened, I don't know, 4.9 million years ago. And so essentially what you end up with when you track these lineages over time and look at where the divergences happen, that is a phylogenetic tree. So here's a cartoon to help you understand that a little bit simpler as an example. So if you look at this, you say, okay, well, that's not quite a family tree. It's a little bit more like this. So yes, I mean, you had a great-grandfather, and so you can imagine you didn't just have a great-grandfather, you had great-grandparents, and a number of them, who, some of them produced your second cousins, as well as you. All right? And then your grandparents, they probably also produced some of your cousins as well as you. And your parents may have produced siblings as well as you. And so you have these branches of lineage that happen over time. And the further back your common lineage is, the more distant the relationship is between you and, say, your siblings you're pretty closely related to, your cousins you're further distantly related to, your second cousins you're even further related to, and you can imagine as you keep going back in time, they're still your ancestors, but the lineages can diverge even further and further apart the longer away you go. Now, it's often, this is a sort of common image that's presented as evolution. Just as this is not your family tree, this is not evolution. This is a better example of it. So you didn't descend from, I don't know, let's say this is some sort of small mammal. They are more like your cousins. You would have had a long time ago a common lineage from some ancestor in which you both descended from that produced these small mammals as well as humans, but you did not descend from those small mammals themselves, but you do share common ancestors to them. Same thing, we did not descend from amphibians. They evolved and we evolved from a common ancestor. Same thing, we did not evolve from fish. Fish evolved from a common ancestor that we also evolved from, that amphibians also evolved from, that small mammals also evolved from. The further back you go in time, the further back these common ancestors can be. So, phylogeny is the study of evolutionary relationships between species, and we tend to draw that out as a phylogenetic tree. So it could look something like this, and we sort of build these on a number of different pieces of evidence. Before it was all visual, structural anatomies and that sort of thing. So dogs look like wolves, so we know they're relatively closely related, and then we can say, okay, well, a red wolf is a slightly different structure to it, and a coyote's much smaller, so they're probably further distantly related to a dog, whereas a gray wolf is relatively closely related to a dog. And so you end up with these farther away connections. And then you can say, okay, well, a fox, I mean, they're quite different, but they would have descended from a common lineage. So this guy right here would have eventually produced dogs as well as foxes. Foxes and dogs, they are related to each other through this common ancestor. So we used to base that on just morphological traits. Developmental structures help out, and we'll look at that in the next slide. Biochemical lineage helps as well, and that's directly related to DNA sequencing and figuring out if we look at the genetic codes of a dog and a wolf and let's say a fox, they share a lot of similarities, but there's more similarities between a dog and a wolf's genetic code than a fox's. So we can use DNA sequencing as more of a conclusive way of seeing how closely related two different species are. Here's an example of how embryonic development can help with this. As animals in particular are developing, they tend to start out quite simply. And so here are a number of different types of animals in different stages of gestation as they are growing. One of them is a cow, one's a rabbit, one's a tortoise, one's a fish, one's a pig, one's a chicken, one's a salamander, and one's a human. But at this particular stage in their embryology, they all look pretty similar. If we look a little bit later in their growth cycle, their distinctions start to become more obvious. But what we can do is we can take a look at this embryological development and say, okay, well, maybe see these guys here look more similar than these two here. So these two are probably more closely related, and these, what is that, 6 are more closely related. And so we can start to get a sense of how embryological development helps us with the morphology that is harder to tell later on in life. And then further on, it is going to get easier and easier to tell, and there's the actual answers. The closer related they are, the more similar they tend to look earlier in development. So, as we've sort of seen before, each intersection in the phylogenetic tree represents a common ancestor. So if we looked at, say, the polar bear and the brown bear, right here is a common ancestor. Kind of hard to see. Right here is a common ancestor that evolved. Some of them evolved into a brown bear, and another group of them evolved into a polar bear. And if we go further back, those guys share a common ancestor with, let's say, the American black bear. So it is further back in time, which shows you the brown bear and the polar bear are more closely related because their common ancestor is more recent in time. And then if we go all the way back to, let's say, let's do the panda bear, the common ancestor between the panda bear and the polar bear is all the way back here. And so we can sort of get an idea of how far back in time these lineages diverged from each other, or how far back in time the common ancestor is. A clade is a smaller taxonomical group that includes a single ancestor species and its descendants. Essentially, this is just a quick way of calling a group something other than a group. So if you had your phylogenetic tree here and you said, okay, I want to talk about this particular group here, instead of just saying this group here, you'd say, oh, well, this clade here. So it's just a particular group within a phylogenetic tree that you have defined to talk about for one reason or another. Cladograms show how organisms are related based on shared derived characteristics. So things like the development of feathers, we could use that as a point on a cladogram to show where that divergence happens. And cladograms and phylogenetic trees, essentially, you can treat them kind of as the same thing, but cladograms should really try to depict time a little bit more accurately. And so you can think of them as being a little bit more of a graph, where they're trying to show time in roughly proportional accurate amounts, whereas a cladogram is not worrying necessarily about their structures maybe per se as much, but more trying to get the time right. We're going to kind of treat them as the same thing. So cladograms, phylogenetic trees, we can deal with them the same thing because we're not actually going to get the accurate measurements of the time either. But what we do want to know is if we have a particular trait. So let's say this trait here, eggshells being developed at this point. So what that means is that this common ancestor here was able to develop the trait of being able to produce eggs with shells. And so the things that evolved from that common ancestor produce eggs with shells. This trait right here is the development of or covering of the body with hair. And so the animals that evolved from that common ancestor, they end up having that trait. Let's go down to four limbs. If four limbs wasn't a thing until this point in time, then all of the members of the clade from that point are going to have that particular trait, or maybe they've lost it in some cases, but they would have had at some point that particular trait. It didn't exist before that point. Cladograms can be drawn as a Venn diagram. So you can imagine if this is your cladogram, that's sort of a drawn out one, it's the same thing as drawing it like this. If you were to look at it from the top view, it would essentially be a Venn diagram. So you can imagine these guys here, that's one circle, and then the B with a C and D in it, because they're related here, would encircle the C and the D as well as the B. And then if you wanted to get the A, the B, the C, and the D in, you'd have to go all the way down to this point here. So you could think of a Venn diagram being like a cladogram, or a cladogram could be drawn like a Venn diagram if you look at it from the top. So try this out. Try making a Venn diagram from the following data. So we've got four groups across the top here, and we've got some different traits with them. So reptiles and birds have waterproof skin. Amphibians, reptiles, and birds have limbs, and fish, amphibians, reptiles, and birds have bones. So you can think of this in terms of a cladogram or a Venn diagram. Let's see if you can do the Venn diagram. So just pause it and see if you can draw it out. Your cladogram would look something like this. This is the point where the fish diverge from the rest of them. So what that means is that has bones, it would have to occur some point before that, because everyone beyond there has bones. If we looked at has limbs, that must have happened some point before we had the branch for amphibians, reptiles, and birds, because everyone beyond that point has limbs. And if we look at waterproof skin, that must have happened somewhere after the amphibians evolved. So the ancestors of reptiles and birds would have evolved that trait. And if we looked at it as a Venn diagram, we could say, okay, well, all of them have bones. We can draw a circle that has all of them in it. But fish are going to be excluded from the circle that would say has limbs, but amphibians, reptiles, and birds would be in that one. But reptiles and birds would have to be inside of their own circle within the amphibian circle. It's within the circle that has fish in it that describes the bones. All right, modern taxonomy. So traditional classification determines relatedness based on morphology. Phylogenetics determines the evolutionary relatedness. And genetics has basically been the way of checking this, to making sure that the connections that we think were there based on morphology are actually true based on the genetics. Because if you're just basing it on just morphology alone, the problem is some of the traits may have changed over time, so it may look like some things are similar, but in fact they are quite evolutionary distant from each other. So let's take a look at this one here. We've got three organisms, and if you look at them, two of them look like they're quite similar, so you would assume based on morphology that maybe they're more similarly related. Now you guys know enough about dolphins to know that that's not true. This is actually basically a type of dinosaur type thing, whereas these guys here are very closely related, both being mammals. So if we looked at a phylogenetic tree, let's do humans, and dolphins would be relatively close related, and dinosaurs would be way, 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 way back in our evolutionary history. If you look at these three, again, two of them look quite similar, but it is the wolf and the tiger that are more closely related. So the Tasmanian tiger would be quite far away on an evolutionary tree. You guys aren't lined up to make the circle very well. But the wolf and the tiger would actually be quite closely related in history because, again, they are both placenta mammals, whereas the Tasmanian tiger is another type of mammal called a marsupial that would have been on its own branch. So if we had a kangaroo in here, it would probably be coming off the branch with the Tasmanian tiger. Again, these things here, we've got two that look relatively similar and one that does not match, but a bat and a cow are quite closely related. Again, they're both mammals, and like the first example, a dinosaur pterodactyl is really, really distantly related. So even though a bat has evolved to have similar characteristics to a pterodactyl because they ended up occupying a similar niche, they both had advantages to taking flight, so they end up with similar structures, but the bat's wings would have evolved somewhere along here. The common ancestor with a bat and a cow would not have had wings, so the bat developed its wings, and the pterodactyl, let's say it somewhere along there, would have also developed its wings separately from the bat. So phylogenetic trees can oversimplify nature. It's nice and easy when you have things like mammals and big organisms like that. They are easy to classify, generally speaking, and there's not a whole lot of crossbreeding with them. So there's not a whole lot of genes moving between different species in mammals and animals in general. That doesn't happen. But that promiscuity between species in something like plants happens quite a bit. So phylogenetic trees for things like plants and definitely things like bacteria or more simple organisms are going to be very, very difficult to do. So if you ever wondered how you get a grapefruit, again, it coming from a plant, there's all of these ways that you can cross these different species to get different results. And so if you look at a grapefruit coming from a pomelo as well as from a line of mandarin oranges, eventually you're able to mix the genes in a particular way that you get a particular trait. So when you try to do a phylogenetic tree with these plants, it's not going to work out as well. So we're going to stick to doing them to easier, straightforward things like animals.